Hello, we're here for the coffee hour. I wanted to fill some people in on things, make sure that they're aware that certain things are going on in the group. We usually use these meetings to catch up on things. It's a more informal meeting time. I don't see anyone here today, but people might come over the course of the hour. So let's take a look at our GitHub repositories. So this is DevoWorm. This is our GitHub presence. We have a bunch of repositories that are pinned. And one of the things we're doing right now is we're doing GSoC 2020. And we have 2 students involved in this. I don't see any pull requests that are active. We've had a bit of activity this week. Mayuk has been adding in a lot of stuff this week. He's been looking at movement in worms. He's also been downloading raw data from the EPIC dataset, which is a dataset where they've captured, they've done cell tracking on a number of embryos, and he's going back to the raw data for that. We use that. We use a processed version of that data for different things, but he's going back to the raw data to get more information about cell movements during development. So that's one of the things that's going on here. Ujwal Singh is also working on integrating the OpenDevoCell resource. So we've got a README file up. He's done a couple of pull requests so far this summer of code. Let's see what we've got here. He has a sort of a roadmap that he's put out. This project will focus on improving the data science and machine learning infrastructure of the DevoWorm group. And so he's got this nice unified model diagram. And so that's nice. I like this. This was in his proposal. And then this is the splash page that he's planning for OpenDevoCell. Let's take a look at this. I don't know if I've really thought about this version too much. Okay, so we've got the splash pages where you present a bunch of pages at once or in sequence. He's calling this DevoWorm AI Center, which is nice because we're kind of branding it in a different way. He's got a bunch of projects up. Devo Methods, DevoWorm Academy. So all that's very nice. History of DevoWorm. The numbers. This is nice. And if you would like to comment on the design in these repos, this is going to go live after the summer of code ends. So in order for you to make comments, if you wish to make comments on the design, I would go to, I believe we set up a project board for this, and I'm not sure if it's here or if it's in the other, I think it might be in the other. So we have another repository for our group meetings, and this is distinct from GSoC. So our group meetings are here. Let's see. Here it is. Our group meetings are in this repository. And well, I don't think we have a separate project for that. Oh, I think I remember what happened. We included it in this project board, but we're using tags. So if you would like to make comments on Ujwal's designs, go to our group meetings repository and go to projects, and you find major tasks for 2020, which is a project board. Go into the project board, and I believe it's in this one. Maybe it's in the other one. Okay, I made out to be a liar here. So it might be in the other. We can go back to GSoC. We'll go back to this later. we'll go to the GSoC 2020, this project board, which is a separate project board specifically for GSoC issues. It's set up exactly the same way the other one is, but it has more specific issues. This is the one. Good. So this one is, we have these labels. So in GitHub, if you want to set up a board, you can go to projects, the projects tab in your repo, and set up a board. And you set it up by putting in columns: to do, hold, in progress, done. Some logical set of categories that you would move the cards around in. Then you put these cards, you define these cards through this plus sign, and you add in a card, and you just name it whatever you want, whatever the issue is. A lot of times you'll name an issue and then you revisit it, and you want to break it into other issues, and that's perfectly fine. Sometimes the issue is not something you ever do, so you put it on hold, and then you may delete it later. And some actually go from to do to in progress to done. So we see we have some done here. But we have these labels, and so this is the way you do this. If you want to comment on Ojwal's layout, you can actually create your own card, which is why I told you how to create a card. And so this one is just the generic design evaluation card. And so if we click on it, we see that there is nothing in there. It's just a design evaluation of Ojwal's layout. So I was planning on having, at the main meeting, some conversation about evaluating the design, people's comments, and so forth. But we want, when you start making these specific design suggestions, I would like you to put labels, this label specifically, in the card. And so how you do this is you open up the card by clicking on the link here at the top of the card, the name. Actually, you have to, when you create this, you have to convert this to an issue. And so I should show you this. So let's create a sample issue here, sample. And you notice it's not an issue yet. It's just a card. You have to go to these little 3 dots up at the top and convert to issue. And then it converts it, and you're done. And I'm not going to convert it right now because I don't want to populate it, but I'll just delete this one. And then once you create the card, you can create them in any category you want. We usually create them in to-do because it's easy to then move it this way. But usually they start in to-do, so it's like, if I think of something that needs to be done in the future, it's always usually in to-do. It might be something in progress if it's something we create sort of as a subdivision of an existing card. But so these labels, I'm going to go back to this and show you how to make a label. So this is the issue. It's pretty generic. We can put in comments here. So we can leave comments. We're going to do that right now. Oh, I've closed the issue instead of closing this. So make sure you don't do that. This is a little wonky. So you actually have to scroll down to cancel this comment if you don't leave anything. Yeah, this whole interface is a little wonky, and you're going to see this with the labels. The labels are right here under assignees, and I can actually assign this to people. So we have people in the group. I think Jesse expressed interest. I'm going to just put a bunch of people on this, so that okay, I'll just put those people on there. And then I'll put DevoWorm as well, which is it's usually me or someone affiliated with the account, but we just put that in there as one of the accounts. So we're just assigning it to everyone, just so everyone can participate. We have other people in the group, but they're usually not in the GitHub. They're usually not collaborators on GitHub, and so that's fine. But this is one way you can contribute to feedback on this. So labels, we go to this wheel, and we say apply labels. Now, this is something that's, every GitHub repository, if you create your own repository and you want to create your own labels, every repository has a certain set of default labels. And so something like bug or documentation or duplicate or enhancement. And these are just things that are typical of like a good first issue, typical of like open source project or coding, where you need to communicate, bring people's attention to things that are, what they represent. So the issue may represent a good first issue for a new collaborator. And so we've used this in trying to attract people into a repository and tell them how to interact with it. This is a good first issue for you if you want to start off with a small issue. It's straightforward. It's not something that's like something we're going to break into different pieces later. It guides people's attention. Things like enhancement or documentation or duplicate, those are things you might use when you want to communicate to people specific things we need in our project. Like if this is a documentation specific issue, or maybe the issue is a duplicate, someone else created another similar issue, or an enhancement in the program or a bug. This helps people see where the trouble spots are, maybe. But we can actually create our own labels, and I've created a label for OpenDevoCell, which is the one I'm going to use for this set of issues, OpenDevoCell being the thing that we want to comment on. Now you can actually create new labels, and this is what we've done here. You can create labels for anything. So say we want to create a label for, it's education related, right? Or it's related to the community period, which we don't actually have a label for, but that's okay. There is a create new labels. So again, like this side is wonky, so sometimes I'll use like, I don't know if you can actually create labels from this part of it. But yeah, so these are the labels. I'm not going to show you how to create new labels right now, but you can create all sorts of labels, and you can even search labels in a very crowded project board. And so this is a digression from the main idea here. If you want to comment on Ojwal's designs, just use if you're a collaborator here, put an issue on the board, label it with OpenDevoCell. Or if you want to see what the issues are, you can search for this tag and you'll find them. And we can, no, we searched. I don't want to search. I want to go back to the main board. Okay. So that's how you do that. And I would say I like the design. Overall, I think it's good. I'm not sure about some of the images. We'll talk about that. The color coding is a bit off. I'm still not. I mean, the color coding is something we're going to probably work on over time, like just to get the right template of colors. I mean, I like the way that there's a splash with different features, but sometimes the features kind of, they're discontinuous if you look at them. Some of the images are, what I'd like to see is actually like maybe a scheme where everything is sort of the same color. Like the images might have like the same color as like a mask. So it's like a transparent mask where you can see through. It's like a green, or what I call DevoWorm green or DevoWorm purple, which is another color we have. And that actually would look a little bit better. And just a little bit, like some of these images are, like it just, the way it's visually jarring sometimes to see like different types of images pop up. Like you have the worm, and then you have this matrix thing, and then you have like cells. It's very discontinuous, but I like the overall design. But that's okay. We can talk about the elements later. The visual elements. So, but I like, this is his general plan for this UML setup, and I think he's coming along pretty well on this. So that's Ujwal. Then if we go here and we go to the okay, so this is actually Pretrained Models. This is our This is Mayok Deb, who's been doing a lot of stuff already with. He's actually got a bunch of blog posts if you're interested to read his progress. He's been doing a lot of stuff with movement. His main goal is to build a pre-trained model for developmental biology and neuroscience. So those embryos that we saw, the idea is to create like a model for the embryos to identify cells and identify other elements, but also meta features, which might be things like movement or other motion or other kind of flows in and around the cells, waves in the embryo. And so those are things we can look at with what I am calling meta features. And I think I have a thing on meta features in here. There's actually a folder or a directory on meta features under definitions and concepts. And we go to meta features. And then we have, so meta features are defined as movement vectors, flows, and expansions. So you see all these things in development. You see the cells, they divide and they move around in the embryo. And they don't just move according to some like haphazard way. They're positioning themselves. And they're also, as they divide, they're splitting their size in two, and then sometimes they expand. The cell expands from that size. So in some embryos, it's a mere, reduction of volume by half. And in other embryos, you have that reduction in volume by half from the mother cell, but the cell grows afterwards. And so we can see this if we look at movies of development that are, continuous representations of people who've acquired images at very, short time intervals. We can see these things unfold. And so the idea is to take all these images and sample, those images as a time series and find these patterns or potential patterns. And so I have a bunch of things that we could look at. I have a link to soap bubble film, which is maybe a nice physical model for embryos and embryogenesis, where so soap bubbles are these. If you take a bar of soap or you could take like if you blow bubbles, there's like they sell those canisters of bubble, of like a film that you dip a wand in and you blow through it and you can create bubbles like a soap bubble. And what happens when they're packed, and I don't know if they show it here. Well, here they kind of show when soap bubbles wet a surface, they have this sort of interface with the surface, and the interface is the bubble packing itself against the surface. And so this happens in a way that's sort of geometrically efficient, energetically efficient, and also has to do with whether the surface is hydrophobic or hydrophilic. In other words, is it something that is, friendly to like liquids, or is it unfriendly to liquids? And there's a bunch of literature in material science on that. But some of these things might be going on in embryos. And I don't see a picture of soap bubble packing, but there is okay, here it is. Several bubbles make a foam. So you can see that bubbles can, when they're a bunch of bubbles in a medium or an interface, they form these patterns. So this looks like a honeycomb built by a bee, but it's not. It's actually soap bubbles that are all packed in some optimal packing. And so they form these patterns. And so these are things that you can characterize with equations. You can we understand how soap packing or soap bubble packing works. There are all sorts of things you can do with that, but so it's one thing we're using for meta features, or at least thinking about in terms of meta features. Another are Lagrangian coherent structures, and so this is another sort of model for flow of things in water. And so Lagrangian coherent structures are sort of if you take a bunch, say if you took like a bunch of particles and you put them into a liquid, and those particles diffused over time, they might diffuse sort of randomly across the surface, or if there are currents in the water, or if there are different, directional forces, like if the particles are actually microorganisms that swim together, you can actually track each of these particles and their trajectory, and they'll actually form structures. So if you take a bunch of particles and you drop them in liquid, if you drop them into this I have a cup of coffee here, right? Let me see if I can get it on the camera. I have this cup of coffee, and it has, obviously a surface of liquid. If I drop a bunch of particles into that liquid and let them diffuse, they'll diffuse and they'll go to the edges of the cup. So you'll have a ring around the edge of the cup of all those particles eventually. Some of them will say, let's assume that they're buoyant, so that they travel along the top of the liquid. So you have this ring of particles eventually. So they all have their own trajectory that they diffuse out along, and you can measure that with the Lagrangian, and then they all end up in this ring. And that ring technically would be a Lagrangian coherent structure. Now, this cup is not turbulent in any way. It's, it's laminar, and so there's no interference with diffusion. But if we have something that's highly turbulent, then that's a different story, and you end up with different patterns of diffusion that are not regular as that ring would be, but they would form different structures, like something like this or something like that, spiral eddies, or different plumes that you might see. Here's a steam ring, which is actually quite regular, but the idea behind Lagrangian coherent structures is this is a very limited description of it, all right? That you use a Lagrangian to track particles, and then they form these patterns over time in the liquid, and it depends on the turbulence of the liquid and what the particles are doing and things like that. So they've used LCSs to characterize physical phenomena in liquids, and coherent crowd patterns in humans and animals. So it's not just for particles necessarily. You can have autonomous agents, if you will, doing things, and you can actually track them. We've actually used this in my other group to look at different representational patterns in human cognition, or in just a cognitive model, or a representational model. And so this is kind of an interesting application to that. But this is another thing that we might look for in terms of meta features. Look for those things. But the first thing we have to do for meta features, before we can get to any of that, is to try to sort of characterize these movement vectors, these flows, expansions. And one way we're going to do that, of course, is to look at just movement in cell tracking data and other types of data. Look at maybe the potential patterns there. There have been papers I think we've reviewed in the group on different forces that are acting upon the embryo during development. We had a paper on hydraulic fracturing, for example, at the interface of cells. So in other words, when cells are in an embryo, there's an interstitial space between the cells, and in those cells you can actually get bubbles forming, or some other set of flows going around the cells, and they shape sort of the morphology and some of the differences that emerge. Like eventually you have this ball of cells that form different patterns, different asymmetries. Why is that? And we have physical models for that, but we don't really have good models from data for that. It's just kind of like people have looked at in different singular experiments and looked at different potential mechanisms. But it's actually kind of unclear of how that process unfolds. And so that's an area that we might be able to move towards with this kind of work. But the pre-trained models idea is actually pretty good. We're not quite. This is another reason why he's getting a lot of this data, and I'm not quite sure Not quite sure, how that's going to turn out. We did a blog post on Pretrained Models last fall. It's on Synthetic Daisy's blog. I think there's a link to it in. I think we may actually have it up on in this DevoWorm ML repository. So if we go in the DevoWorm ML repository, Pretrained Models, we do have a link of the blog post. So this is markdown version of the blog post. And here it is. So it's actually was put on both Synthetic Daisy's blog, which is my own blog, and the Node, which is a blog run by the Developmental company. And this was published last fall. And it's about 1,500 words. We had a bunch of people in the DevoWorm ML group who contributed to this. And so the argument here is that you can use pre-trained models that are specific to developmental biology. So another reason why Myoke is developing this pre-trained model is because we want a model that will analyze just things specific to developmental biology. So specific to a system where you have cell divisions, for example, or you have things that are changing their form or shape often. And so those, if you think about regular pre-trained deep learning models, for example, like I guess Deep Lab is one example. They really deal in you might call them mechanical objects or objects that are static over time. So if I train my model on a bunch of bicycles or street signs or faces, they have their own dynamics. Bicycles and faces. We usually take static images and train the model. But there are special challenges to developmental biological data. And actually in medical imaging analysis, we have a similar problem where, you have specific types of features you want to discover. And it's very hard to do using something like bounding boxes or using training your model on, a bunch of, irises, which are parts of the eye, or bicycles, or numbers and letters. It just isn't the same quality of shape, I guess is the best way to put it. It's really kind of hard to describe unless you've kind of been exposed to developmental data. And so this was a vision at the time, and we laid it out, and then we proposed a GSoC project. So this is the sort of the result of that. And so we actually, since that time, and I don't know if I have it in here, I think it's back in this repository, the GSoC repository. People have actually created some pre-trained models for, I don't know if it's here or not. People have created some pre-trained models for different parts of the cell. So there's a group, and I wanted to bring up the links, but I can't seem to find it right now, where people have looked at different parts of the cell, like the mitochondria or some other part of membranes. So these models are basically trained on images where, it presents a bunch of membranes or a bunch of organelles, and it trains a model to segment those pieces of the cell. So it's, again, you have very specific pre-trained models to biology. Maybe not to what we want, but definitely there are people who are working on that outside of our group. And so I can't seem to find the links to these resources. Oh, let's see, maybe it's in here. Well, I don't know. I think that's not the one I was thinking of. It could be in the definitions and concepts. No, it was just there, so I don't think that's it. Okay, so why don't I, well, if you root around these directories, you'll probably find the links. I'm not going to get into it any more than that, but let me go over some more of the definitions and concepts. So we have meta features, and then we have something called polysemy, which is, so Polysemy means that a feature can have one of many meanings. So polysemy means that if you have an object, if I go to different people and I ask them what it means, that it will have different meanings to different people. If I take a painting, like an abstract painting especially, and ask people, what does this mean to you? And people will give you different answers. The meaning, the content, information content of the painting, and the sort of the physical attributes of the painting, color of the paint, and the orientation of the lines. Those things are kind of separate from the meaning. They're related to the meaning, but they're not separate. And so polysemy just means that, unlike the physical sort of invariance of the object, we don't really know. People actually perceive. Physical invariance. People, they derive their meaning from the fact that they look at certain parts of the image, or maybe the thing in its entirety, and then, so the physical invariance is there, but it's not necessarily the way people interpret it. But the meaning is, can be variable across individuals. And so we're trying to develop this idea of, and through some of the semantic machine learning models that we're applying to the problem here. And we've been trying to work on this for a while, and it's really kind of elusive, because when people talk about semantic content in computer science, they often mean just labeling things, right? We're trying to go a little bit farther than that. So in this case, let's go back to the definition here. For example, a single feature could have a series of possible meanings. So we want to have some context for something, and then those meanings are actually the context of the image, such as C cell, oval, left anterior, neuron nodes. So these are kind of like the kind of labels that we typically think of when we associate semantic content. But we think about this as maybe a single feature having multiple meanings, and then relating those meanings together in some way. So I'm trying to bridge the gap between sort of what is technically feasible and sort of the larger idea of polysemy, which is this idea that's highly subjective, but also, if we can figure out how to capture it, it would be great. So this can be done using labels derived from annotations, but we can also use more sophisticated labels derived from beliefs and cultural values. So the biological equivalent to this would be the role of a cell in a larger tissue or organism, or characterize a membership in a gene expression profile. So C cell, for example, this feature that or this label that we have, has a 45% membership in expression profile Y. And this is misspelled, but let me fix it really quickly. But what that just means is that you have, say you have some feature of the data set or of the data, and this label isn't uniformly applicable to it. In other words, if I have a gene expression profile and I have a bunch of cells, gene expression profiles may be highly variable, especially a profile that has multiple genes in it. I can divide that expression profile up a bit by these multiple labels. So in other words, I can take all of the cells that are associated with that gene expression profile, and then I can take that gene expression profile and break it down into components. So one component would have one label, one component would have another label, and so forth. And so you can think about this in terms of membership degree. And so this is actually a concept from fuzzy logic or fuzzy learning, where you take an object and you say, how much of a membership does it have in this set? So if it's 45% membership in the set, that means it's 45% it's present maybe 45% of the time, or is maybe 45. I don't want to talk in probabilities, because that's where it gets a little fuzzy, but pardon the pun. But this 45% membership idea, if you actually go through and you walk through an analysis, you'll find that they have a membership function and the membership function determines their degree of association with something. So they use a terminology of membership, but it just means like how to the degree it's associated with something. And so the idea would be that you'd have these partially associated labels with some. It could be a cell, it could be a gene expression profile. I'm just using expression profile as something to put operational cast on it, but it could be anything. It could be a group of cells, or a developmental period. So, if it's during this period of gastrulation, how often does this configuration hold? The things like that. So that's the idea behind polysemi. I have to update this description because it's not really that clear what we want. I think I've thought about it a little bit more, and other people have as well, and so we'll be updating that soon. Semantics, we don't have anything for that, but that's forthcoming. If you'd like to contribute to this, you can go into this Definitions and Concepts tab. So that's our GSoC, and then our group meetings, we have a couple things to go over. So let's go over our group meetings real quick. We have a bunch of different directories here. These are just things that have happened in the group that we've kind of thrown up here. There's community support, which is more like about the actual community of people who meet. We don't have anything under that, but we're probably going to in the coming weeks and months. Biological imaging is a very generic topic that we often talk about. Complexity measures is something that, let's see, what's in here? I don't even know. Oh, this is where we're looking at different. So this is a topic that we are thinking of exploring, a couple of us, in terms of like understanding measures of complexity, complex behavior, complex systems, what have you. And the idea here is to create maybe create complexity measures, or to evaluate them. Or I think in this case we're thinking about visualizing them. And one thing that I had an example from years ago, which was this bread vector model, which they use in ensemble forecasting. They use it for climate change. They use it for weather prediction. But they have these. This is basically just a sort of a boxes and arrows diagram of a model and how you would think about these different models and measures. And so this is just a way of describing it without too much math. There's a symbol here, but that just tells you that you're integrating. And that's and then this is so like the idea would be to create a bunch of different visualizations of these models and measures, to create our own models and measures. We had a talk by George Mikhailovsky a couple weeks ago on his new paper on complexification, and I should put that in the README here, a link to that paper. But it's a really good paper. He goes through this idea of sort of entropy and information theory that's really kind of, I think, useful to a wide range of systems in biology, maybe even outside of biology. But he talks about this idea of complexification. And it's really interesting, some of the linkages he's made in this paper. I was thinking about it the other night, and I'm not prepared to talk about what I was thinking about, but I think it's very applicable to a lot of different questions that we're grappling with in the group. And so this is our complexity measures repository. If you'd like to contribute, it's a little early now to think. I mean, probably maybe just to make a comment. And again, if you want to make a comment on any of this, you go into the projects board, the project board, and we have a different project board for this. This is major tasks for 2020. And we just have like a large number of issues here. A lot of these are finished. A lot of these are in progress. Some are related to, like this is probably revitalized today's meeting. I mean, that's more or less done, but also in progress. This one I think presented on last week, so that goes into finished. And this is the complexity measures issue, so you can actually comment on this issue if you want, or you can issue a pull request to that repo. There are a lot of things you can do here. Always interact. So a lot of things to do in the group meeting with respect to the group meetings. Let's go back to group meetings. Let's go through these folders a little bit, and then I'm going to get to one of the papers we're working on. We have data science and structuredness. I can't remember what this was, but that's another topic. Developmental brains and perception. That's something that's forthcoming. Where I talk about morphogenetic agents, which are something that we have some slides on here. So what we do here in some of the repos is we take a bunch of like PowerPoint slides, upload them as images, and then put me in the README. So you can go to the README and actually have this little slideshow where we have things that are, these are older versions of this slideshow, but it basically describes the idea. And this is an idea where we have these agents that are both observers of patterns and emitters of patterns. And the observer has like a perceptual substrate, and the emitter has a morphogenetic substrate. So the emitter can form patterns like this pattern here, a checkerboard, or like, it's not really a French flag, but it's kind of like the idea of a French flag. And then the perceiver actually tries to figure out what that pattern is. So you can emit a pattern as an emitter, and a receiver will pick up the pattern and try to decipher it. And agent can actually have two of these components at the same time. So you can have, it's basically a link between creating spatially restricted shapes and attentional mechanisms. We can model sort of like coevolutionary relationships where, you have a predator-prey type relationship where the prey is constantly creating new patterns to hide from the predator. The predator is trying to decipher those patterns. Or you can even create a situation where there's feedback between pattern formation and perception of pattern formation. So if the morphogenetic part of this is generating these weird patterns, weird sort of, shapes that are, or patterns that are something that's never been seen in nature before, that helps with perception. That can sharpen the attentional resources of the perceiver. And so this is kind of an interesting model both for developmental biology and for like agent-based perception, agent-based cognitive science type stuff. So this is a nice, I'm going to present on this in a couple weeks maybe. We have developmental physics, which we talked about earlier. We don't have anything in that repo or in that folder. DevoZoo, which is of course the data that we have. This is where I was putting some of the plans to restructure the resource, but we're trying to move into a couple directions. We'd like to add in molecular data to our pantheon. We have a lot of morphological data, but we would like to have more molecular data in the way we have, in the style we have it resourced. We'd like to adhere to FAIR standards more. So FAIR standards are standards for data sharing, findable, reusable, interoperable, and I can't remember what A stands for. But it's basically making sure that people can reuse your data and having community standards for that. So that's something maybe more on the technical side, but if you're interested in data sharing, we should talk about that. Also focusing on the integration between data sets and metadata. So, we're on the quest for more metadata. And so the semantic stuff that I presented earlier, or talked about earlier, is actually part of that metadata quest. And so if you're interested in that, let me know as well. So to wrap it up, 15 minutes left in this meeting. Why don't we talk about a paper or a set of papers that we've been trying to do? So we've got this. If you go to this digital Basileria project, we've done a lot of stuff with this Basileria, which is a diatom. It's a marine microorganism. And we've done a lot of stuff with morphology and trying to digitize that using deep learning and biomechanical models to kind of get at this, trying to digitize an organism, basically. So this is kind of what they've done with OpenWorm in terms of C. elegans. We're trying to do a similar thing with diatoms and specifically Basileria. And so we put out a preprint on this, which is going to be a book chapter. This is actually, if you read the README here, you see a Basileria. It's a marine microorganism. It has these filaments that move against one another, and it's like an accordion. It moves back and forth through the water column, and it exhibits different, stopping behaviors and stretching behaviors. And people don't really know why. There are like at least 20 models of how it happens, perhaps, or people proposed about 20 models for this, none of which are necessarily totally satisfying. But we don't really actually know why that happens. So we did this paper on analyzing the morphology. And in the process, we ended up thinking about behaviors and how they're generated. And so we have two papers we're working on here. One on Basileria psychophysics and one on Basileria COPGs. And what are those? Well, it's a good question. First of all, psychophysics may seem a little absurd for a marine microorganism, but why don't we talk about that first? So we're proposing here that, basically Basileria colonies generate behavior, and they do this and the behavior of the colonies is driven by actin, filaments and other things that induce sort of excitable behavior, which is not really a brain, but it's doing information processing and it's behaving. So it's responding maybe to the water column, it's responding to some sort of autonomous drive. We don't know exactly what it is, but we can basically model it sort of like a brain. Use a metaphor to say it's a brain. It exhibits a sort of non-neuronal cognition, so it doesn't have a brain, and yet it's exhibiting behavior. And what how like a brain is it? And so we use this idea of psychophysics, which people have done in bacteria, by the way. This isn't just something we're pulling out of a hole somewhere. This is something that's been done. And so this model of cognition results in the production of behaviors related to phototaxis and psychophysics, which can be distinguished from random behavior using a series of criteria. And so this paper basically, sort of deals with this set of issues. So there's a hypothesis number one, which is that there's Hebbian learning, perhaps. Each filament in the colony exhibits sliding motion. Sliding motion is coupled by neighboring filaments, and it results in a rhythmic oscillation that occurs in multiple time scales. And so there's also entrainment over time. So all those things, maybe we want to apply a model of Hebbian learning to see if that's a good model for describing what's going on. And again, Hebbian learning maybe is an incorrect description of what that model would be. There's a model called, I think, the Kuramoto oscillator model. There are also excitable oscillator models that we could use instead of Hebbian learning, or to contrast Hebbian learning with those things. Kuramoto model is used to describe firefly behavior, because if you ever watched fireflies in the grass, they fly up above the grass and they flash their little luciferase. They have a luciferase sac in their tail, and it gets excited by electrical pulses, and there's a light source. That's why they call them fireflies. Well, it turns out these fireflies can synchronize their behavior across fireflies. And to do that, they've used these kind of oscillator models to describe that synchronization process. Because the idea is that they fire sort of an electrical potential every so often, and it results in luciferase being activated. And so you see these flashes of light. And so they're rhythmic, but they're unsynchronized, unless they're synchronized by collective behavior. And so that's the idea here. And Hebbian learning is because you don't really have, you have a certain type of collective behavior. It's sort of like a serial collective behavior where you have filaments that are aligned serially. So they're not there's no parallelism here at all. But so this is hypothesis 1. Hypothesis 2 then is that this is seemingly neuronal behavior. Can we develop a Turing test for distinguishing between like a neuronal generated versus a non-neuronal generated behavior? And this has something to do, there are a couple of people have addressed this sort of informally. A-Life 2019 had a fake life recognition contest, which was like, they would build these generative models where there would be organismal or agent behavior, and they would try to determine whether it was like, how real it was. Or even in OpenWorm, we've looked at models generated in the platform Geppetto, and then compared them to real worms to see, which ones are simulated, which ones are real. Can you detect the difference? And with C. elegans, you have a lot of stereotype behavior, so maybe it is hard to determine the difference. And so this is a table that kind of talks about neuronal versus non-neuronal instances of behavior, and then behaviors generated by those processes. And this is kind of a sloppy typology of that. So this is still probably in progress. I would say it's in progress. If you have ideas, you can issue a pull request here or comment in the project board. That would be appreciated. The other paper is the COPGs. So what are COPGs? So to understand that, we have to go to the idea of central pattern generators. And so this is the idea that those basilaria that are moving, this is sort of focusing on a specific hypothesis, which is that those rhythmic movement patterns are not central pattern generators themselves, but similar to that concept. So a central pattern generator is like the group of neurons that are wired together in a network, and they generate oscillations. So it's some small neural circuit. So like you have pacemaker neurons in the heart, or in some other, in some organisms they use them for, well, like in fireflies or in electric fishes, where they use them to generate a pacemaker or something that's rhythmic, firing that's rhythmic. And so central pattern generators generate this pattern. It's autonomous. It doesn't require any thinking or input from other things. It's just that little neural circuit making a pattern. And so, but this requires excitable cells, neurons. We don't have excitable cells per se here. We have cells that have maybe some excitability, but they don't fire action potential. So in Basilaria, we observe something I call collective pattern generators, which are COPGs. So this is where you have collectives of cells that are generating a pattern, and they're generated from collective and coordinated behavior. So the Basilaria phenotype, you can see those filaments that are stacked like that. They're exhibiting coordinated behaviors, but no central nervous system or brain is involved. And so we go through the neural architecture of this, and there's some modeling that's involved, just basically modeling those oscillations of sine waves, looking at some of the, what happens if you delay different parts of the chain in terms of sine waves. So you have delayed sine waves. So like they move in pairs, they slide against one another. One cell will slide against another cell, and that will be a pair of cells sliding like this. But then there's a second and third cell, and a third and fourth cell do the same thing. So they end up sliding against one another, and they can straighten out like this, or they can move together like this, So it's like an accordion where you're pulling it apart and together, and then the parts of the accordion, the folds of the accordion are moving, but they don't all move in parallel. They move serially. So one affects its neighbor, the neighbor affects its next neighbor, and so forth. And you get this sort of, like you call it a chain reaction, but it's actually probably not that. So we can model those chains by a bunch of sine waves that are out of phase, and you might get some harmonics there in terms of the oscillator. We can also see spindles at the extreme of each cycle, and then you can model this as either like just a sine wave that goes on indefinitely and then slows down when it stops, or it can stretch out and stay there. So that's where the sine wave goes to a maxima and then stays at the maximum. And so we have a bunch of graphs in here that show these simulations, kind of describing this, boil these oscillations down on a tractor map, and then I have a sine wave with random noise. So this is a sort of a representation of these spindles, but we've done it a little bit more. We've given it a little bit more noise here. So we're trying to analyze this in different ways using some tools from dynamical systems, and then maybe applying some algorithmic information theory to it. And so I'm hopeful that this paper is going to work out pretty well. It's going to be a nice paper, but I don't know exactly what it's going to look like. So again, if you want to contribute to this, you're welcome to as well. So thank you for attending the meeting. We're almost at the top of the hour. We didn't get anyone else in, but I wanted to go over some projects for people to see what we're up to. Definitely had some coffee here, as you saw earlier. We have our group meeting in a different location on Mondays. And so check our website out, DevoWorm.weebly.com, for more information on that, the link, the meeting time. And if you're interested in presenting either in this meeting or the Monday meeting, let me know. Okay.